Okay, good. And I actually don't have any announcements for today. And so I will turn you over to Dr. Nidich, who will be speaking for the full session today on the topic of bacteria of the GI. So I believe he's going to do, it's probably lecture for about an hour or so, maybe a little less. We'll take a short break and then he'll finish up. All right, Matt?
Sounds great. Thank you, Ian. And I should tell everybody that they're welcome to interrupt me at any time throughout the lectures, right? Just
Right. So yeah,
unmute.
sorry, I was going to announce that. I forgot already. Yeah. So I know, as you know, I normally monitor the chat, but as I say today, because we're not getting a huge number of questions. I think it would be fine if you had a question to ask. It might be more timely for you just to wait for a convenient moment and unmute yourself and go ahead and ask the question directly. That seemed to work just fine last week when I was lecturing. So let's try that again today. All right. So we won't use the chat box today. Just if you have a question of Dr. Nidich, just unmute and go ahead and ask. All right.
All right. I'm going to share my screen here. Let's see. Pen. All right. So again, my name is
Matt, I just wanted to confirm, are you recording? I don't see you recording.
I sure am not, so I'm glad you mentioned that. Yeah, so for some reason my ability to record disappeared when I went full screen. Here we go. Please ask the host to give you permission to record.
Make hosts allow to record local. You know what? I'm not even seeing that as an option, which is really strange. Let me make you a co-host. It looks like you're recording now. Okay, good.
Am I?
So you don't even have to do it. By making you a co-host, that automatically makes you a recorder if I'm a recorder.
Co-recorder. Okay. Awesome. All right. You can see my PowerPoint slide, yes, Ian?
Yes, everything's fine.
All right. Good afternoon, everybody. Once again, my name is Matthew Nidich. I'm an associate professor in the Department of Microbiology, Biochemistry, and Molecular Genetics. Feel free to email me if you have any questions, but I think you'll find these both parts. So there are two parts in this, sort of one longer lecture. I think you'll find them very accessible, the material very accessible, but if you have any questions, please feel free to reach out or, talk to each other first, preferably. I think that's a useful exercise, but then, of course, if there's still a question, email me. Today, I'll be talking about bacterial diseases of the GI tract. So I'd like to help you with your studying for the exam. My questions are very straightforward. They come only from these slides and the things I say in the lecture, no place else. And I would like to help you by telling you what the outline is for lectures 1 and 2. So the first, let me see. The first lecture will have a background on GI pathogens and generally, how these diseases are transmitted, that sort of thing, the effects on society a little bit, just general background. And then we're only going to talk about 2 different bacteria in the first lecture. One is called Vibrio cholerae, causes a disease called cholera. And then we'll talk about enterotoxigenic E. coli, which causes traveler's diarrhea. It's one of the main causes of traveler's diarrhea, and we'll talk about what that is. But in the important distinction between those 2 bugs in lecture number 1, and then the bacteria that we'll talk about in lecture number 2, is that those two bugs, Vibrio cholerae and Enterotoxigenic E. coli, cause diarrhea, not dysentery. So what is dysentery? Dysentery, simply put, is bloody diarrhea, okay? And part two, we'll have a brief background, and then we'll jump into a few bugs that cause dysentery, namely Shigella, enterohemorrhagic E. coli, and Campylobacter and Salmonella species. Then we'll talk about a type of Salmonella that causes a systemic infection called Salmonella typhi. It causes typhoid fever. And then we'll finish by talking about bacteria called Helicobacter, or Helicobacter pylori more specifically, that causes ulcers and in some cases can lead to cancer, okay? So, but all of these are pathogens of the gastrointestinal tract, okay? And feel free to interrupt me if you have any questions. Okay. So you can read these objectives on your own. Let's just jump into it here. Okay, so foodborne illnesses are widespread. In the US, there are at least 48 million people per year that acquire foodborne illnesses. 128,000 or more are hospitalized, and believe it or not, about 3,000 people die.
Matt, I'm sorry to interrupt. I got the impression that you had advanced your slides, but no, but I don't think we're not seeing your slides advancing.
Oh, you're kidding. What do you see right now?
Just that first slide. It says bacterial diseases of the GI tract, part 1.
You're kidding. I've seen my, is it moving now?
Nope.
You're kidding me. It says I'm screen sharing. I updated my Zoom before we, let me try re-sharing the screen. Thank you for that. That's a new problem. Do you see it? Do you see it now?
Nope.
My goodness.
So
All
did
right.
you unshare and reshare?
I'm going to do that right now. I'm going to share something else, and then I'm going to share the PowerPoint again.
All right.
Please tell me you see facts on foodborne illness now.
Nope, don't see anything. Anybody else seeing anything other than their first slide?
Yeah.
No, nothing.
No, we're not seeing anything.
Okay,
Nope.
let me I wonder if that's something to do with the recording. How about now? Still no?
Right now, I see you're just starting the screen sharing, so it hasn't come up yet.
Also, I'm not even on Wi-Fi. I'm on like hardline straight into
Yeah.
my,
Yeah, this is strange. I've never seen this happen before. So
oh my goodness. I'm gonna
you know,
Should
we
I-
can we'll take a minute. We'll try to get it going here. If not, Matt, you can just
let me
do you have a PDF version? You could try that.
let me dump out of Zoom and come back in real fast, okay?
Okay.
Any better?
All right. So I see the PowerPoint.
Okay. I'm gonna hit
All right. Go to full screen.
record again. Oh, it's asking me for you to give me permission again, but all right.
Oh, there we go. We're good.
All right. Should I try recording again or?
Yeah, let me just hang on a second. I'll do that.
Okay.
All right, you're recording. All right, I think we're good to go.
Okay. Why it won't let me? I see the problem. Boy. Give me 2 seconds. I think I see what the problem is here. All right, so yeah, there are about 3,000 people that actually die of foodborne illnesses in the US every year. Ian, just so you know, oh yeah, now I'm recording again. Okay, you must have given me co-host abilities. So it's actually so while adults older than 50 years old suffer the most hospitalizations and deaths, it's actually very small children under 4 years who are sickened by food more than those in any other age group. And there are many examples of foodborne illness, foodborne contamination. There was a sort of ripped from the headline story in 2019 where salmonella contaminated uh, a number of peanut and peanut-related peanut products or bags of peanuts, things like that, and peanut-related products. It was actually 3,600 different products that needed to be recalled. This was from a company in Georgia called the Peanut Corporation of America, and it actually led to over 714 people being infected in 46 states. The ages of the people affected were less than 1 year old to 98 years old. And whenever we hear about these foodborne illnesses or outbreaks, these are always underreported. You know, a lot of people don't go to the doctor or the hospital if they're sick, so these are always underreported. But in this case, 23% of the people that were infected with salmonella from these peanuts or peanut-related products were actually hospitalized. It's a very severe outbreak. Half of the people affected were children, and this actually resulted in 9 deaths. The really remarkable thing about the story is that the company knew that they were shipping contaminated products, and they attempted to cover it up before, during, and after the outbreak. And there were 2 brothers that owned this company, and it was so egregious what they did. And there were email records, you know, of them trying to cover this up, and you know, arguing that they had to ship the products even though they knew it was contaminated. That one of them ended up with a sentence of 28 years in prison, and the other 20 years. Okay, so there are other examples of foods being, you know, contaminated. In 2010, there was a very large salmonella outbreak that led to the recall of 550 million eggs, so more than 500 million eggs being recalled from one company in Iowa. There were 2,000 reported illnesses, and like I said, these illnesses go underreported. In 2017, there was a recall of contaminated soy nut butter. And the one thing that made this one notable, I thought, was that the name of the company that you know, distributed these contaminated products, resulting in this large recall, including 25-pound pails of soy nut butter, is called I Am Healthy. So they are not, as it turns out, as healthy as they thought. So I'd like to show you another example. Let me just share something different here. Let's see. New share. Yes. So right before Thanksgiving, a few years back, there was this warning that came from the FDA and actually the CDC in this case, after 32 people in 11 states fell sick with a virulent form of E. coli. They said the contaminated food was romaine lettuce, and the warning was that if you did not know whether your lettuce was romaine or whether a salad mix that you had contained romaine lettuce, do not eat it, just throw it out. And they say that the tainted lettuce was grown or processed in Canada, in the United States, and that's a common theme with these things. We don't know exactly always where the contaminated food is exactly coming from, at least initially. There was this like nationwide warning. It was like, a really big deal because it led to the hospitalization of 13 people, including one person who developed kidney failure. And the reason for that was that this type of E. coli is the Shiga toxin producing E. coli, known as enterohemorrhagic E. coli, and we'll go into that a lot in the second lecture. And it had the designation O157H7, and we'll talk about what that designation means when it comes to E. coli. One amusing thing from this article was that they quoted this guy, Steve Felberg, who was apparently standing in his kitchen in Montclair, New Jersey, preparing dinner, and he felt stymied. He pored over the lettuce he bought over the weekend at the local farmer's market. And he said, I think it's romaine, but it's not classic romaine. So we're taking a picture and emailing it to the hydroponic farm to find out before we eat it. So that's what we're up against here, is that, people are told, your lettuce might kill you or make you paralyzed, which EHEC can do, or cause you to lose your kidneys, requiring a kidney transplant, because that's what enterohemorrhagic E. coli can do: kill you, paralyze you, cause you to need a kidney transplant. Yet people are still stubborn enough that they don't want to throw out their lettuce because they're not exactly sure whether it's the type that that the CDC is saying is contaminated. All right, so back to the PowerPoint. So while developing nations still have a lot of trouble with these infectious GI pathogens that cause infectious diarrhea, where it's a major cause of illness and death, especially in infants and young children, in developed nations like ours, it's not a huge problem leading to death typically, but it is second only to the common cold for causing us to lose time, in productivity, doing the things that we'd like to do rather than, laid up with diarrhea, right? So it's actually very easy to control the spread of most of these pathogens, which is why they're not such a problem in developed nations like ours, because if you provide clean drinking water, properly dispose of sewage, and slaughter and process meat, poultry, and seafood in a sanitary way, it's not much of a problem. And what I've also listed here is balanced food supplies. And what I mean there is that when you are hungry or starving, you don't have proper nutrition because you're malnourished, your immune system suffers, and you become more susceptible to these pathogens that cause diarrhea. And again, that's a problem in developing nations for sure. So one common theme is that these pathogens are transmitted via the fecal oral route. So there's some contaminated feces from humans or livestock in particular, and their feces contaminates our food or our water supply, and then we either ingest it or get it on our hands and then touch our hands to our mouths and become infected with these bacteria. And this is a very important point. All the bacteria that I'm going to talk about today require the bacteria to grow, okay? So they must be ingested and survive and grow in our gut in order to cause disease, and we'll talk more about that. But we ingest these organisms, and I said sometimes we're also ingesting their toxins at the same time, but this is in contrast to some other bacteria that you've learned about that produce a preformed toxin that makes us sick, and the bacteria don't necessarily need to grow to cause us to become very ill. And all the bacteria that I'm going to talk about today, the bacteria must colonize our body, our gut, and then grow. So they grow, and they typically produce some kind of toxin or virulence factor, and then maybe they remain in the GI tract, where they would cause diarrhea, and then become excreted into the environment, and then start this process all over again. Alternatively, instead of staying localized to the GI tract, they can move through our epithelia and become a systemic disease, and we'll talk about that later, especially when it comes to Salmonella typhi, causes typhoid fever, for example, which then is typhoid fever, even though it's a systemic disease, is also a diarrheal disease or a dysentery that where the pathogen Salmonella typhi is excreted, and then again escapes into the environment to contaminate our water supply or food supply, and start this process all over again by contaminating another person, infecting another person, okay? But we have remarkably good defenses against GI pathogens. In our mouth, we have our normal flora coating the epithelia, preventing the bacteria that we ingest from necessarily attaching to our epithelia, and we have enzymes in our mouths that attack the bacteria. For example, lysozyme, that is an enzyme that degrades the peptidoglycan cell wall that all bacteria have. And then in our GI tract, we have the epithelia shedding. So there's like a turnover of the epithelia that line our GI tract. And then even if some bacteria would attach and start to colonize, well, maybe they attach to a cell that sloughs off, and the bacteria just get a ride on the epithelia right out of our bodies before they get a chance to set up shop and cause us problems. So we have special immune cells in our GI tract called the MALT, or mucosal associated lymphoid tissue. We have bile. Like I said, we have enzymes such as lysozyme, and we have our immunoglobulins, antibodies that are the cell-mediated immune response against a bacteria. Most bacteria do not like bile, but we'll talk about, again, Salmonella typhi in the second lecture, that can live indefinitely in bile, that loves bile. So it can set up shop in our gallbladder forever. And then we have our normal flora, lining our whole GI tract. Like I said, the bacteria that we ingest have to compete with that normal flora for an attachment site. And then also don't forget that our stomach has a very low pH, as low as 1. And the bacteria have to survive that low pH environment in order to set up shop in our GI tract. So we'll talk about some bacteria that are remarkably good at surviving low pH, and so that you only need to be infected by a few of those bacteria. They have a very low infectious dose, 50. So then maybe only 50 of those bacteria are required to cause us disease. That's a coincidence that their LD50 just happens to be 50, but it could have been any other number. But anyway, that's a very small number of cells that would be necessary to cause disease in that case. Just want to make sure you guys can see me here. Okay. So, like I mentioned, we have, as long as our mucosa is intact, and we have mucus forming and liquids flowing, saliva flowing from our mouth through our intestines, and we have this peristalsis, which is the motility, our gut motility, pushing all of the things through our GI tract. It's not that easy for bacteria to cause us disease. These GI pathogens. Typically we need a large inoculum before we become ill. I just want to point out that there are cells in our intestines called M cells. So here's an M cell, and M stands for microfold. And these cells are not professional antigen presenting cells like, say, a macrophage, but what they do is they coax the bacteria into them, and then they pass the bacteria from the luminal side of the intestine into the tissues, where the bacteria, once it passes through the microfold cell, can then be transferred to a macrophage, a professional antigen presenting cell, which can then process the bacteria and display antigens to T cells, and then where you then can get a B cell response, so cell mediated response, immune response, and then the production of antibodies, and these antibodies can be coupled to what's called the secretory factor. And then the IgA coupled to the secretory factor is then secreted into the luminal side of the intestine, where you can get this antibody response in the luminal side where the bacteria are colonizing. Okay, so you have these very elaborate systems in place to deal with this constant onslaught of the bacterial GI pathogens. All right, so like I said, all the bacteria that I'm going to talk to you about today require colonization or growth of the organism in the GI tract. So this means the bacteria have to be living, right? And we'll talk about a bunch of these, but just some examples include Salmonella, or some of the diseases caused by these bacteria include Salmonella, gastroenteritis, bacillary dysentery, which we'll talk about in the second lecture, and cholera, which we'll talk about in just a moment. And this is in contrast to intoxication, which does not require the presence of living bacteria in your gut. It's the ingestion of a preformed exotoxin, and some examples include staph and Clostridia, which causes botulism, and we won't be talking about those today. So, like I said, in the first lecture, we'll be talking about watery diarrhea. Then the second lecture, we'll be talking about bloody diarrhea, which is also called dysentery. We'll finish that lecture by talking about one type of bacteria that causes not just dysentery, but it's a systemic disease. Again, that's Salmonella typhi. And then we'll finish up finally by talking about Helicobacter pylori, which causes peptic ulcers, okay? But all the bacteria that I'm going to talk to you today belong to two different families, and this is really going to help you when it comes to studying. They either belong to the Enterobacteriaceae family or the Vibrio-Naceae family, and I'll talk to you more in a second about what that means. If you start to Google around a lot, you'll find that two of the bacteria that we're going to talk about, Campylobacter and Helicobacter, now belong to their own families. The Campylobacteriaceae and the Helicobacteriaceae, but up until very recently, Campylobacter and Helicobacter were part of the Vibrio-Naceae family because they really fit the bill. But then with all the genetic sequencing and everything, there were some arguments made that they belong in their own family. But for the sake of studying, I encourage you to think of all the bacteria as fitting into one of these two families. Okay, and we'll talk about that as we go on. So, all right, in the first lecture, we're going to talk about diarrhea. Diarrhea is a non-inflammatory disorder, so there's no pathogenesis, no severe pathogenesis in the GI tract when the bacteria are there. It's primarily a disease of the small intestine, leading to fluid and electrolyte loss. And like I said, there's very little histopathology detectable because they're not really doing a terrible damage to the GI tract. There's often an enterotoxin involved. We'll talk about these toxins in just a moment, but diarrhea can be very mild, known as the runs, to something very severe, sometimes fatal disease, and we'll talk about that. Like cholera, for example, is a very severe diarrhea. There's no blood involved, no dysentery involved when it comes to cholera, but it's just a diarrhea, but it will kill you if you're not properly treated. Now, dysentery, on the other hand, is an inflammatory disorder, primarily of the large intestine, sometimes of the small intestine as well. It involves invasion of the colonic wall, and there's often remarkable histopathology because there's epithelial cell death, ulceration of the mucosa, acute inflammation, and then that's where the blood and the pus in the feces comes from. It's from this damage to the colon. So there's usually some sort of toxin involved, and then pain, fever, cramps, nothing good comes from having dysentery. Okay, so like I said, all the bacteria that we'll talk about today belong to the Enterobacteriaceae or Vibrio families. All the bacteria that I'll talk about today are gram negative, okay? So the gram stain is not diagnostic since all the bacteria I'm going to talk to you about are gram negative. However, the Enterobacteriaceae are straight rods, gram negative straight rods, while the Vibrio are gram negative curved rods. And these are things that we can observe just by putting the bacteria in a light microscope. You can see whether they're straight rods or curved rods. All the bacteria I'm going to talk to you about today are also non-spore forming. Okay, so the Enterobacteriaceae form a capsule, sometimes known as a slime layer. It's this outer coating, while Vibrio do not have this capsule. Both the Enterobacteriaceae and the Vibrio are found in the intestines of mammals, but Vibrio can also be found living free in seawater or in shrimp and other seafood. Specifically, the Vibrio cholera is what I'm talking about in this case. It's halophilic, it likes salt. Okay, so Enterobacteriaceae can be motile or non-motile, while the Vibrio are highly motile, having these very impressive flagella that enable them to swim through our GI tract also, and get to a spot in our body where they're happy. So like outside of the stomach, for example, where the pH is not so low. All these bacteria have appendages that enable them to attach to the host cells. We call these pili typically, and we speciate these. That is, we determine what bacteria it is that we're dealing with using clever diagnostic tests. Some of these are special agar plates that have different reagents in them, such as lactose or maybe sorbitol or whatever, and we look for their ability to grow on lactose or sorbitol because that's diagnostic of what type of bacteria it is, and we'll talk a lot about that. We also use these surface antigens called the O and the H antigen, and we'll talk about that in just a minute as well. So, okay, so these are just some examples of enteric diseases caused by the Enterobacteriaceae. E. coli and Salmonella cause diarrhea, while Shigella and E. coli, different species can cause dysentery. We're not going to talk about Yersinia, but that is an important one, but we're not going to have time to talk about it. Salmonella typhi causes typhoid fever, and E. coli can cause hemorrhagic colitis, EHEC, for example, and we'll talk about that in the second lecture. While Vibrio family members like Vibrio cholerae cause cholera, Vibrio parahemolyticus causes gastroenteritis, Campylobacter jejuni, we'll talk about that one, causes diarrhea and dysentery. Helicobacter pylori causes peptic ulcers, we'll talk about that one. And then here's another Vibrio vinificus, like similar to Vibrio cholerae and Vibrio parahemolyticus, causes gastroenteritis, but this is also a common wound infection, especially in fishermen. So Vibrio vinificus lives in the ocean, and we'll often find that fishermen who are exposed to Vibrio vinificus maybe have have diarrhea, but then will also maybe have an infected wound. So it's not uncommon to get hit with that double whammy as well. All right, so there are many determinants of pathogenicity in these two families. So there's the endotoxins, the LPS, the lipid A portion of the cell wall, which causes which causes a toxicity, fever, fatal shock, and other changes. We think of fever, especially when it comes to endotoxin. But endotoxin is not so much a cause of diarrhea. Okay, so in that case, we're talking about the enterotoxins produced by the bacteria that cause the transduction of fluid into the lumen, like in the intestines. And these enterotoxins are the causes of things like traveler's diarrhea and cholera. We'll talk about that. These bacteria often have flagella, as I mentioned. So many of them are highly motile, so they can swim through our GI tract, set up shop. But we'll talk about one bacterium that has no flagella. And these bacteria all have the ability to grow on or in our mucosal cells, and then cause disruption of their function, disturbance of their function or cell destruction in the case of the dysentery causing bacteria. All right, so there are numerous cell surface components that are important for a variety of reasons, but in this case, they're very important because we use them in diagnostic tests to determine what type of bacteria we're dealing with. So there are specific antibodies using these diagnostic tools or tests to these cell surface antigens. And one of them is the capsular antigen called the K antigen. And it's called the K antigen because it was discovered by German scientists, and the word for capsule in German starts with the letter K. So it's just the capsular antigen. So these are these mucopolysaccharides on the outside of the bacteria. There's the LPS, the O antigen, which is the outer portion of the LPS. And again, this is not necessarily the cause of diarrhea, but it is a cause of fever, and in some bacteria, and it's also a target for these diagnostic assays. And then there's another one called the H antigen, which is an antigen on the bacteria flagella. And the reason it's called the H antigen is because in German, the word for breath, like you're breathing, breath, is Hausch, and the H antigen is also called the breath antigen because the scientists who discovered it before, like 100 years ago, before the advent of molecular tools, noticed that some bacteria, when they were pipetted onto a plate, would form a haze on the plate, and that haze reminded them of the condensation that forms on a cool piece of glass when you breathe your breath onto it. So they called the bacteria that could form that haze, that's due to the bacteria swimming, they called that, they said that those bacteria had the H antigen, this flagellar antigen. Okay. So, like I said, all the bacteria that I'm going to talk about are gram-negative, so the gram stain is not diagnostic. We use carbohydrate fermentation tests, such as their ability to grow on lactose, or their ability to grow on special media. We'll talk about that, or in, say, low oxygen conditions. We'll talk about that. As I mentioned, we have these diagnostic tools, serology to the O, H, and K antigens. And we can use, very old-fashioned assays where we look for agglutination of these antigens in stool samples, just using antibodies to these antigens, or the antibodies coupled to certain things like beads to these, then it would call, when the antibodies are mixed with the cells, they cause this clumping, and then you see this like precipitate form. But there's much fancier diagnostics as well. We'll talk about some of those. We look for the presence or absence of specific enzymes. We'll talk about that when it comes to things like Helicobacter pylori, for example. But now, we live in this, modern age where everything is PCR based, and we, have very sensitive, very specific diagnostics for all the bacteria that I'm going to talk about today. We're using the bacteria's genetic sequence to identify it. All right, so now I'm going to talk about the first bacteria that we're to cover in depth, and that bacteria is called Vibrio cholerae, and it causes cholera. So in 2010, there was an earthquake in Haiti that initially killed 220,000 people. This was, disaster of biblical proportions. And to make matters worse, by the end of the same year, there was a cholera outbreak that directly resulted from the response to this earthquake. By November, cholera spread to the entire country. And by December, there were over 121,000 cases and, close to 3,000 deaths, actually. So this is, I'm sure, underreported. By the middle of the next year, there were over 420,000 cases, 6,000 deaths. A year later, 7,000 dead, 500,000 cases, 5% of the population affected. And more recent numbers are something like 8% of the population was affected. But what made this particularly remarkable is that Haiti did not have cholera prior to this earthquake. Now, where this came from is the Nepalese delegation from the UN that was sent to Haiti to help with the recovery did not properly dispose of their feces at an encampment. They were dumping it into a river. And in Nepal, where this delegation was from, Vibrio cholerae is endemic, and they managed to bring their endemic strains of Vibrio cholerae to Haiti and infect the entire country just from that one campsite. So now, in large part due to some vaccines that were developed in the last few years, this outbreak in Haiti is well under control, thankfully. But what you can see here are a bunch of bottles surrounding this patient. And these are all the bottles of electrolyte that were required for this one patient to survive one bout of cholera. And cholera is the most severe diarrheal disease to affect humans. It's transmitted in contaminated water, and some people believe it could also be transmitted between bodies of water by insects. It can be transmitted in infected food, such as shrimp and seafood, and not just in our bathing water, but in our drinking water and well water as well. It's very easily prevented by treating sewage adequately, not dumping it directly into the river, and water purification systems can easily remove cholera, Vibrio cholerae. And if we avoid raw or undercooked shellfish as well, you're not going to get cholera from seafood. There are short-term vaccines that are very effective, which is basically the only reason that Haiti has recovered from this outbreak. Not the only reason, but in large part due to the effectiveness of these vaccines. And untreated, the fatality rate is over 60%. But in fact, it's quite straightforward to treat cholera, and we'll talk about that in a second. But you can see how severe it is. In this child here, their skin is tenting. So what tenting is, and this is called cholera gravis or tenting. So when you have cholera, you're so dehydrated that your skin loses its elasticity, and then if you pinch it, it sort of stays in the shape that it was pinched in. So that's called tenting. That's how severe the dehydration is associated with cholera. But probably more than anything else, what saved people from cholera over the last many decades is something called a cholera cot. And a cholera cot is simply a folding bed that has a hole in it, and that hole is covered with a plastic tarp, and that plastic tarp has a sleeve in it, and that sleeve leads to a bucket. And what happens is the patient's feces are collected, the diarrhea is collected into this bucket, and there's a ruler in the bucket that measures the amount of fluid loss by the patient. So the healthcare provider knows exactly how much oral and IV rehydration to provide to the patient. And that alone is often sufficient to save the patient, as long as they come in for treatment before things get too far. And if they do, antibiotics can be used, but really antibiotics are not necessary to treat cholera. Oral and IV rehydration is typically sufficient. Okay, so cholera, Vibrio cholerae, to cause cholera, must be ingested in large numbers, okay? So really, to get cholera, you need to ingest about 1/2 ml of a stationary phase culture. And that's a lot of bacteria, a lot of bacteria. So most labs that I know that work on cholera grow cholera in the lab almost like it's a laboratory strain of E. coli, because they know that if they get splashed with cholera, they're not going to, or you get, the worst case scenario, something you're like splashed in the face with cholera or something crazy, which, really doesn't happen. But even if it happens, you're not going to get cholera, because you'd have to drink it to get it, basically, because cholera is very sensitive to our stomach acid, which is probably why we need such a large inoculum to get cholera. So it has to cause disease, it has to colonize the small intestine, and this requires the bacteria to be motile. And cholera also produces this enzyme called mucinase that enables it to drill through the mucus layer, and then it has to attach to receptors in our gut and produce a toxin. And we'll talk about the cholera toxin in just a moment. But all of this causes a massive loss of fluid and electrolytes. But the important thing is, this is not a dysentery. There's no blood or white blood cells in the stool. This is simply diarrhea. And what I pictured here is a villus. This is just to remind you that in our GI tract, we have these goblet cells that are producing mucus, okay? And the bacteria need to drill through this mucus layer, okay, that's surrounding the villi in our intestines, in order to gain access to the cellular epithelia pictured here, all right? So those goblet cells are pumping out this mucus that's acting as a protective barrier in our gut. All right, so now I'm going to tell you about cholera toxin, and cholera toxin is extraordinarily well studied. We understand the mechanism of its action, very, very well. And it's the archetype of a family of toxins that are found not just in cholera, but in many bacteria. So it's a structurally similar protein found in many different bacteria that cause diarrhea. And I'm going to tell you now how this works. So normally in our cells, we have this protein called adenylate cyclase here, and it produces cyclic AMP. Our cells only require a very, very small amount of cyclic AMP to function properly. And cyclic AMP is only produced by the adenylate cyclase when the adenylate cyclase, this enzyme here in blue, in its kidney shape, is bound to another protein called a G protein. And if that G protein is bound to GTP, okay? Now, the reason the cyclase only produces a small amount of cyclic AMP is that this G protein quickly hydrolyzes GTP to GDP. And when this G protein is bound to GDP, not GTP, it does not interact tightly with the cyclase, okay? And the proteins dissociate. And when they're dissociated, the cyclase does not produce cyclic AMP, okay? So it's this hydrolysis of GTP to GDP by the G protein that keeps the cyclase in this very, low activity state. Now, what happens when we have a cholera infection is that the cholera toxin uses NAD to ADP ribosylate the G protein, okay? So when this G protein is ADP ribosylated, the GTP is no longer hydrolyzed to GTP, and the G protein stays in the GTP bound form, and it stays very stably bound to this adenylate cyclase, and you get a ton of cyclic AMP produced in your cells. So in a normal functioning epithelial cell, we have the tissue bringing in water and sodium, potassium into the tissues, okay? In a cholera infected cell, we have the opposite. And what happens is there's a transporter, a chloride transporter in the membrane. And what happens when cyclic AMP levels are high is that the cyclic AMP activates a kinase. Okay, it's not important what kinase that is, but that kinase phosphorylates, P for phosphorylation, this channel. And when this channel is phosphorylated, it stays open and chloride goes rushing out. Following the chloride is water. Okay, water follows the chloride out of the tissues and into the lumen. And the sodium and the potassium follow the water. So when we think about diarrhea and electrolyte loss that happens when we have a diarrheal infection, what we're really talking about is this opening of the chloride channel and the fluid flux and electrolyte flux from the epithelia and into the lumen. Okay. So I'm going to use this next slide. I'm going to take a diversion on this next slide, okay? Because the cholera toxin is so well studied, we understand its mechanism so well, it is a perfect opportunity to talk to you about what's called regulation by translational control. So here is the cholera toxin. It has two proteins in it. There are 5 copies of the B subunit, the binding subunit, 3, 4, 5 B subunits. These are the binding subunits, and 1 copy of the activity subunit. And this oligomeric complex binds to a target on the epithelia here. I want to ask you to know what this target is. And then this complex sits down on the cell, and then once it sits down on the epithelia, then the activity subunit is transported into the cell where it causes the opening of the chloride channel like I described. All right. Now, the interesting thing is that there are 5 B or binding subunits and 1 activity subunit. However, the genes encoding for the A subunit and B subunit are in an operon in the bacteria where there's 1 promoter driving the transcription of these 2 genes. So here's gene A, here's gene B, and there's 1 promoter, okay? So if there's 1 promoter driving the transcription of these 2 genes, how is it that we end up with so much more B in the cell than A? In fact, we have about 7 times more B in the cell than A. Well, it turns out that there's another level of control going on here, and that is translational control. So the ribosome binding site for the B subunit is much stronger than the ribosome binding site for the A subunit. And because of that, we have a lot more translation of the B subunit than the A subunit. Okay, so that these are the B subunits, these are the A's, and it's because of the activity of the ribosomes at this ribosome binding site compared to this one. All right, so if anyone asks you, how is it that we have so much more B subunit than A subunit if the two genes are encoded together in an operon that's driven by a single promoter? Well, the answer is translational control. Okay, so back to Vibrio cholerae. If we look at an electron micrograph of Vibrio cholerae, we see that they have this curved shape. They look like fuzzy mice, right? So you see the curved shape, long tail, flagella, okay? So these are curved rods, very important to remember. And we can identify cholera on plates that we include a reagent called thiocitrate bile salt sucrose. And if we include that reagent, then the colonies will appear yellow on these plates. Vibrio cholerae are oxidase positive. That means that they have an enzyme that enables them to use oxygen as a terminal electron acceptor. And there's a reagent, the very complicated name that I want to ask you to remember, that turns this like purpley color if bacteria are oxidase positive and we include it in their growth media. We also have serological tests for Vibrio cholerae to the O antigen, or more commonly these days, we just use PCR to various genes in cholera to identify. Okay, so all right, so Ian, I don't know if you're there. I just want to take like a 1 minute break here, see if anybody has any questions, and take a sip of water. If nobody has any questions, I'll just keep moving.
Okay, so why don't we, open, anybody has any questions, now's a good time. I'll leave the recording on for.
Okay.
All
We
right.
haven't finished with the first lecture yet, but there's only a few slides left in the first one.
All right, so it's 20, what is it? It's, where are we?
257. So there's
We'll start.
there's only a very small number of slides, so I'll just keep moving here.
Okay, that's fine.
We can stop for the next one. So that's all I'm going to say about cholerae. Now we're going to talk about enterotoxigenic E. coli. So a professor of microbiology attended a meeting held in a rural part of Mexico. She was careful to consume only well-cooked meats and vegetables and drink bottled water until the last day of the conference. As the heat and humidity became unbearable, she drank a glass of Coke, in which the waiter had added ice cubes. Painful abdominal cramps and voluminous watery diarrhea developed the next day and continued throughout her overnight flight back to New Jersey. After her arrival home, she phoned her physician, who concluded from her travel history and symptoms that she had traveler's diarrhea. The physician recommended oral rehydration and prescribed ciprofloxacin. The antibiotic was completely effective in eliminating her symptoms. So when you travel to a part of the world or even another part of the United States in some cases, where you are not used to the endemic bacteria of that region, and you eat some food or drink some water from that region and you become sick, mainly from enterotoxigenic E. coli, that's prevalent in certain regions, you get what's called traveler's diarrhea. And the CDC's guidance for traveling to regions where you don't normally go is when you're eating, to boil it, peel it, cook it, or forget it. So traveler's diarrhea is watery diarrhea. It's not dysentery, no blood. Often there's vomiting and cramps. And again, it's caused by differences between our normal flora and those of the region that we're traveling to. It's a self-limiting disease, it just goes away on its own over time. Typically, you can take antibiotics, but you don't have to. But often when we talk about traveler's diarrhea, what we're talking about is enterotoxigenic E. coli. And the way to remember this is that the T in ETEC, enterotoxigenic E. coli, the T is for traveler. And there are a number of ETEC toxins, One is the heat labile toxin, LT for labile toxin. And this toxin activates adenylate cyclase. It functions much like the cholera toxin, increasing cyclic AMP, leading to fluid loss. And we call that the labile toxin because this toxin, much like the cholera toxin, is a full-size protein, right? So it's a globular protein, and if you heat it up, it unfolds, and if it doesn't, have its three-dimensional structure, it loses its activity. That's in contrast to the heat stable, ST, stable toxin, toxins found in ETEC. And these are a family of small peptides, okay? And the reason that they're stable is that they're small peptides, so their shape is not as important for their function as their just their sequence, which is they're small peptides. They maintain their shape even when heated, partly because they're full of cysteines that form disulfide bonds. And disulfide bond, as you should know, is a covalent attachment, covalent bond, and you could not break up a disulfide bond with heat. So these ST toxins there's many of them, and a lot of them activate guanylate cyclases, causing an increase in cyclic GMP. I won't ask you to understand how those work, but they work kind of like the cholera toxin that activates cyclic AMP to, but in this case, increasing cyclic GMP, resulting in fluid loss and electrolyte loss. And one thing is that these toxins in ETEC are carried on plasmids, okay? So we always worry that these plasmids are going to jump from ETEC to another E. coli or even into another species of bacteria. So I mentioned previously that we use carbohydrate fermentation tests to identify bacteria. The most famous of these is the McConkey agar lactose plate. So we take this agar, McConkey agar, and McConkey agar, the classic recipe for McConkey agar has lactose in it, and bacteria that form a pink colony on a McConkey agar plate did so because they were able to use lactose as a carbon source, and we refer to those as lactose positive, as a lactose positive result. And sometimes we say that bacteria is lactose positive. E. coli are oxidase negative. They cannot use oxygen as a terminal electron acceptor. And I can identify what strain of E. coli we're dealing with using the O and the H antigen. And what I've pictured here is what's called an API strip, or analytical profile index strip, which is a miniaturized laboratory test. And what you do is you culture the bacteria that you want to identify, you grow it up in some media overnight, and then you add a little bit of that culture to each one of these cupules. And in each cupule, I don't know, there's like 20 of them here, each one has a different reagent in it. And depending on the result observed in each cupule, you look in a book and you look for the result that you observed, like yellow, orange, whatever these colors are, clear, green, precipitate, clear, and then you just check it off, like, cupule 1 is yellow, cupule 2 is yellow or orange, whatever, 3, etc. And then depending on the result in these 20 cupules, you can pretty unambiguously identify the species of bacteria that you cultured. Okay, so this first cupule here actually is the one that contains the reagent that turns yellow when bacteria can use lactose as a carbon source. All right, so ignore this. I didn't give any handouts. There's no table 2. Just ignore that. But I just want you to know that there are other agents of watery diarrhea, E. coli, not just enterotoxigenic E. coli, There's enteropathogenic E. coli, enteroadherent E. coli, diffusely adherent E. coli. I just don't have time to talk about all the different E. coli virotypes, but enterotoxigenic E. coli, ETEC, is not the only one that causes watery diarrhea. In fact, there are many other types of E. coli in general. Commonly, we think of them as 7 virotypes: ETEC, which we talked about, enterotoxigenic E. coli. There's enteropathogenic E. coli. You can remember the P is for pediatric and not just pathogenic. So the EPEC is a diarrheal causing disease that in children mainly, and then young infants, it can cause death even from diarrhea. Enter-invasive E. coli, enter-hemorrhagic E. coli, EHEC, we'll focus on that a lot in the next lecture. Enter-adherent E. coli, diffusely adherent E. coli, uropathogenic E. coli. You might be interested to know that uropathogenic E. coli, as the name suggests, is responsible for UTIs. But the difference between these strains is the presence of virulence factors, often present in plasmids, but sometimes on the genome itself. But these are all E. coli, the same species, just they've just picked up different virulence factors. Okay, so we're out of time for the first lecture, so I'll skip going over the key concepts. But in general, know that these are transmitted by the fecal-oral route. You can simplify your studying by knowing that they belong to the Enterobacteriaceae or Vibrio families. They have to attach to a surface, colonize our gut to grow. They often have toxins that cause the fluid flux, and we can treat everything that I've described for you so far. We can treat with just oral rehydration therapy or IV rehydration therapy, but if necessary, we can use antibiotics as well. All right, so let me go over this question, example question, very quickly. Something like this you might see on a test. In fact, this might be even a little more complicated than what you'd see on a test. My test questions are very straightforward. There's no ambiguity. I'm not trying to trick you. So a 25-year-old woman presents to the ER with a 2-day history of severe diarrhea and vomiting. Okay, diarrhea, not dysentery, diarrhea. Her symptoms started shortly after she returned from a trip to a rural area in Central Africa. Okay, so she's traveling outside of where she normally lives. She recalls eating shrimp that seemed undercooked. Her symptoms started abruptly with watery diarrhea, followed by vomiting. She has not had fever or abdominal pain. On examination, her temperature is 37.2, which is normal. Her pulse is 115. Blood pressure is 80/50. Her mucous membranes are dry and her eyes appear sunken, so she's dehydrated from the diarrhea. Her stool is watery and tests negative for blood. So again, this is a diarrhea, not dysentery. Stool culture reveals a gram-negative curved rod. Okay, what is the major virulence factor that contributes to her symptoms? All right, so gram-negative, that doesn't help us. Everything I told you about is gram-negative. Curved rod, okay. So now we're talking about a Vibrio family member, and it's diarrhea, not dysentery. So I've only told you about one type of bacteria that's a Vibrio family member that causes diarrhea, not dysentery, and that is Vibrio cholerae. Okay, so what is the major virulence factor of cholerae? Well, you're probably thinking already, cholera toxin, and you'd be right. So cholera toxin is the correct answer. It's not the heat labile toxin, that's ETEC. It's not LPS, that's not the major virulence factor of cholera toxin that is contributing to the symptoms that she has. She doesn't even have a fever. Capsule, nope. Flagella, well, yeah, I mean, Vibrio has flagella, but that's not the major virulence factor. The superior answer would be cholera toxin, and I hope that's obvious to you. All right, so let's take a 5-minute break, and then we'll pick up with the second lecture.
Sure, it's 3:09 right now. Why don't we start up again at 3:15?
That's perfect. Thank you.
Can we ask
I
questions?
said, I think I said, I don't remember saying it was more dangerous, but I think that the thing that we worry about when these virulence factors are on plasmids is that they can be, transferred to other bacteria, right? These are do you know what a plasmid is?
Pardon?
Do you know what a plasmid is?
I'm kind of shaky on it.
Okay, so plasmids are episomes. So they're genetic elements that are separate from the genome. Okay, they can replicate in the cell and be and be carried to daughter cells, and they often carry antibiotic resistance or some virulence factor, but they're a major cause of like, I don't know if you've ever heard the expression horizontal gene transfer, but it's a way that genes can be transferred from one species to the next. And we use them in the lab as genetic tools to move genes around and for the overexpression of genes and proteins in cells. So it's like a small movable genetic element.
Okay, thank you.
You're welcome.
Professor, I had a quick question too. So, on the same slide. So when you're talking about heat, label, is that how it's pronounced?
Yeah, that's fine.
Heat stable, is that just like a characteristic of the toxin or how it functions in a host organism? Like,
Yeah, great question. Yeah, it's not a function of how it functions. It's just a I use function. It's not a characteristic of how it functions. It's just a characteristic of its structure. So it, again, the heat labile toxins are sensitive to heat because it's a full protein, right? It's a globular protein whose three-dimensional shape is very important and required for its function, while the heat stable family of toxins are just these small peptides. So, a couple dozen amino acids or less. And you can heat them up and then they kind of maintain their shape, or they just refold very quickly into their normal shape. It's just a way of characterizing them. They also have a lot of disulfide bonds, and disulfide bonds are heat resistant. So yeah, it has nothing to do with how they function in the patient, right? It's just a characteristic of the fact that they're small and contain disulfide bonds, or large and generally don't.
okay, thank you.
You're welcome.
Professor, I was wondering if you could go over the slide that you're talking about, the cholera toxin mechanism of action, really quickly. Just what happens when the cholera toxin comes into play, with the G protein.
So which part of the slide were you asking about?
This second portion.
This part?
Where and where there's like cholera toxin in the cycle plus NAD, and like what happens with that. I didn't catch.
Oh, here? Okay, so in order, all right, I'm just gonna have to basically reteach this again. So adenylate cyclase, you understand, is only active, that's this enzyme here that produces cyclic AMP, right? Adenylate cyclase is only active, producing cyclic AMP, when it's bound to the G protein, and when the G protein is bound to GTP. Okay.
Yep, got that.
So what happens is normally G proteins hydrolyze GTP to phosphate and GTP and GDP very quickly. And that's just inherent property of that they have, is that they hydrolyze GTP to GDP. And what that does is that makes this interaction between the cyclase and the G protein very transient. Okay, so you only get a small amount of time when cyclic AMP is being produced. Now, what the cholera toxin does is it takes NAD and it uses it to ADP ribosylate this G protein, okay? So this G protein becomes modified by the toxin. And when it's modified, it no longer hydrolyzes GTP to GDP. The G protein stays bound to this cyclase, and the cyclase stays active, producing cyclic AMP. All right.
Okay,
Any
got
other
it.
questions?
Thank you.
You're welcome. All right. Need. All right, let's keep moving here. In the second part, we'll be talking about bacteria that cause dysentery. And if you really take a deep dive into the literature, you'll see all these different descriptions of bloody diarrhea, hemorrhagic colitis, bloody diarrhea, dysentery. Don't worry about the distinctions between them. Just know that dysentery is bloody diarrhea. And then we'll be talking about a couple different type of bacteria that cause dysentery, including Shigella. We'll talk about enterohemorrhagic E. coli, Salmonella, Campylobacter, a few others. We're not going to talk about enteroinvasive E. coli. It looks so much like Shigella that there's no point in talking about it separately. But let's get started. Let's talk about the first one. We're going to talk about Shigella, which causes what's called bacillary dysentery. And this is a shigellosis because the genus is called Shigella, shigellosis. And it's a dysentery that affects only humans. It's transmitted by our fingers and flies and in our food and from our feces, again, via the fecal oral route. It affects the large intestine and it's confined to the GI tract. This is not a systemic disease. In places like daycare centers and in large institutions that house a lot of people, such as nursing homes, maybe a prison or even a cruise ship, this is one of the main types of bacteria causing diseases or dysentery causing diseases, bacillary dysentery. And if you maybe you know in daycare centers these days where there are very young children in diapers, the daycare workers wear gloves when they're changing diapers. That's like 100% due to the fact that Shigella is very easily transmitted by contaminated hands. So the incubation period is about 2 to 4 days, after which the infected person will have severe abdominal cramps and fever, and anything ranging from a mild diarrhea to commonly a severe dysentery. And this is stools that contain blood and mucus as a result of the damage to the colon. We can diagnose this type of bacteria using stool culture, and we can treat patients with simple rehydration, but also antibiotics. It's easily prevented if we're careful about washing our hands, properly disposing sewage, and chlorinating water actually prevents the spread of Shigella species. So there are 4 major species of Shigella: Sonii and Flexneri, that are endemic in the US. There's a third one called Boydii that's very rare here. And then there's a fourth one called Dysenteriae that really is responsible for most of the deadly epidemics that we hear about, especially those on cruise ships. Okay, we use the O antigen to speciate Shigella species. We do not use the H antigen because Shigella do not have flagella. They are non-motile. There is no H antigen to look for because they do not have the H antigen. They do not have flagella. So most Shigella species are lac minus. You can just think of Shigella as lac minus. There are a few species of Shigella that pop up from time to time that are weakly lac positive. That is, if you grew them on McConkey agar for a very long time, they would start to appear pink. They could maybe use lactose as a carbon source to a very small degree, but really I want you to think of Shigella species as being lac minus, so they'd be uncolored or white on McConkey agar plates. What's very important is that Shigella and Salmonella are very challenging to tell apart, except for the fact that Shigella form green colonies on plates that contain a reagent called hectoin. Okay, so they form green colonies rather than black colonies because Shigella does not produce hydrogen gas. Salmonella, which we'll talk about briefly, does produce hydrogen gas, and that hydrogen gas reacts with the hectoin reagent and forms these black precipitates due to the hectoin reagent. Okay. Shigella are oxidase negative. Okay, so Shigella, for it to cause disease in our colon, has to survive the stomach acid, and, first pass through the small intestine. Eventually, it attaches to the colon. You can see it attaching, and then it has virulence factors that affect the epithelial cells and cause the epithelia to bring the Shigella inside of them. Okay, so the epithelia import the Shigella using phagocytosis. Okay, then the Shigella dissolve this vacuole that the epithelial cell used to phagocytose the Shigella. The Shigella multiply like crazy inside the cell, and then they can move to neighboring cells where they would do the same thing, multiply like crazy. And then they have virulence factors that cause the cells to become necrotic and die. Okay. And it's that damage to the cells in the colon that lead to the blood and the mucus and the pus in the diarrhea, or in this case, dysentery. Now, the reason that Shigella is so contagious, and the reason that daycare workers have to wear gloves when changing diapers, for example, is unknown. But the ID 50, the infectious dose 50 for Shigella, is coincidentally about 50. So 50 bacteria or cells are required for the patient to become sick. This might be due to its acid resistance. That is, it can survive the acidity of the stomach. It can grow inside of cells, like I just described. We don't really understand why it's so infectious, but these are reasons that people are looking at. So the virulence factors that enable Shigella to attach to the colonic epithelia and then cause the epithelia to take up the Shigella and then dissolve the vacuole and then multiply inside the cell. A lot of those are encoded on plasmids, virulence plasmids, and we refer to those as the invasion plasmid antigen gene products. So the plasmid is the invasion plasmid, and then the genes on there are the invasion plasmid antigen gene products. Okay, and those are very important for the pathogenicity of Shigella. And then there's a toxin that is similar to the cholera toxin in that it has 5 binding subunits and 1 activity subunit. But this toxin has evolved not to modify G protein, but rather to modify the ribosome, okay, the eukaryotic host ribosome. And this toxin, called the Shiga toxin, inactivates the ribosome. And when it shuts down the ribosome, now there's no protein synthesis in the cell, and this results in cell death. Okay, so that's where the cell that's where the cell destruction comes from: the Shiga toxin and the invasion plasma antigen gene products. All right, so that's all I'm going to say about Shigella. Now we're going to talk about enterohemorrhagic E. coli, or EHEC. So in this one, like, highly profiled or publicized story from 2011, there were scientists at a conference in France who became very sick, and around the same time, people in Germany were getting sick, very sick, and not only did they have dysentery, but they were having, in the most severe cases, kidney failure. And this struck 4,000 people, and eventually, actually 54 people died. And there were 850 cases of this syndrome called hemolytic uremic syndrome. That's this kidney damaging disease. And we'll talk about what that is more in just a few minutes. But this was caused by a strain of E. coli called enterohemorrhagic E. coli 0104H4. I'll explain what that means in just a moment. There was a previous example of an EHEC outbreak in 1993 in Seattle, Washington. And what happened in that case was that dozens of young people were showing up in the emergency room at local hospitals in Seattle with kidney failure. And it's highly unusual, I'm sure you know, for children, teenagers to have kidney failure. But the one thing that all these young people had in common was that they had recently eaten at a Jack in the Box or a Wendy's. And what they had been infected with is E. coli strain 0157H7. And this outbreak eventually struck, and then there was a separate outbreak in 2007 of the same 0157H7 that struck 41 states. And what happened here, so like in this case, at the Jack in the Box restaurants and Wendy's, it was contaminated hamburger meat. So we often call EHEC disease hamburger syndrome. And there was this other case in 41 states in 2007 where there was another outbreak of 0157H7, and this outbreak was profiled in the New York Times. So here's an example of this woman, Stephanie Smith. She was 22 when this article was written in 2009. So she was 20 years old when she was paralyzed after being stricken by E. coli in 2007. And sorry, I'm having trouble with my Zoom here. And it said, officials traced the E. coli to hamburger her family had eaten. So she ate an undercooked hamburger at a backyard barbecue. Okay. So what all these people had who were, so I should say, so she was paralyzed, right? So not just were her kidneys affected, but she was paralyzed. And I'll explain why that happens. But what causes this is, again, enterohemorrhagic E. coli, which causes hemorrhagic colitis or hamburger syndrome. And the way to remember that EHEC causes hamburger syndrome is that the H in EHEC stands for hamburger. And it's caused by us eating contaminated or raw, undercooked or raw contaminated ground meat, raw milk, fruits or veggies, or contaminated water that has EHEC. It causes abdominal cramps, diarrhea, fever, and or vomiting may or may not occur. Okay, but the main thing is that there's copious bloody diarrhea, otherwise known as dysentery. And the predominant serotype in North America, especially the United States, is 0157H7. In Europe, it's 0104H4. Okay, so this is the O antigen 157, which is at the outer portion of the LPS antigen designation 157, and the flagellar antigen the house antigen number 7. And this again is the one we find in the United States. And what happens is EHEC attaches to the colon, colonizes the colon, and there's a production of Shiga toxin. Okay, that Shiga toxin, the one that can shut down the ribosome function in our cells, that toxin can move across in between our cells and get into our bloodstream, and it can lead to the death of our kidney cells and the cells of our central nervous system. Okay, and the reason that this happens is that the receptor for this Shiga toxin on our epithelia is in high concentration on our kidneys and our CNS. Okay, so when that toxin attacks our central nervous system, that's when we become paralyzed, and when it attacks the cells of our kidneys, that's when we end up with this hemolytic uremic syndrome, and then we lose our kidneys. And this is an acute disease, renal failure, and it's life-threatening. And the patient requires kidney dialysis and often a kidney transplant. So this is extremely serious. Now, you might be thinking, well, if they have E. coli and it's this serious, I'll just give the patient antibiotics and, kill the E. coli, no problem. Well, big problem. If you give antibiotics to someone who has an EHEC infection, you will basically guarantee that they will need a kidney transplant, or that they'll be paralyzed, or that they'll die. Because what happens is that E. coli has what's called an SOS response. It responds to environmental stress by upregulating and downregulating certain genes. And one gene that it massively upregulates in response to antibiotic stress is the Shiga toxin. So while you may ultimately kill the E. coli using the antibiotic, you will not kill the E. coli before it has produced enough Shiga toxin to kill the patient. So you do not prescribe antibiotics to someone who has an EHEC infection. We identify E. coli, enterohemorrhagic E. coli, by growing them on what are called SMAC plates, or sorbitol McConkey agar plates, where the lactose has been substituted with sorbitol, because most E. coli are sorbitol positive. That is, they'll appear pink on sorbitol containing McConkey plates. EHEC, however, is sorbitol minus. The colonies will be white on a sorbitol McConkey agar plate, okay, in contrast to most other types of E. coli that will appear pink. We have antibodies that can tell us exactly which type of EHEC we're dealing with, say 0157 or 0104, H4, H7, that kind of thing. We can use ELISAs to detect the Shiga toxins in the stool, and we just use PCR very commonly these days. And I already explained to you why you never use antibiotics to treat EHEC. All right, so now I'm going to talk about Campylobacter. So Campylobacter enterocolitis is the most common cause of bacterial diarrheal illness in the US. For some reason, people don't know about it the way they know about Salmonella and E. coli, but it's incredibly common. 1.5 million cases per year, causes 500 deaths. Mainly it affects children and young adults, transmitted via the fecal-oral route, like all of these things. It's a commensal in the intestines of many animals, particularly cattle and chickens, and their eggs. Most of the cases are caused by Campylobacter jejuni. The remainder are caused by Campylobacter coli. Its pathogenesis is very similar to Shigella. Invasion of the intestinal epithelia. It produces a heat labile toxin like the Shiga toxin. So this is a full-size protein, like the Shiga toxin present in, say, Shigella or EHEC, like we just learned about. Only a small inoculum is required to cause Campylobacter enterocolitis, maybe 1,000 organisms at the most. Interestingly, it can colonize both the small and the large intestine, not just the colon. Okay. It causes an acute inflammation of in the small and large intestine within a week of ingesting it, causes a fever, headache, aches and pains, cramps, diarrhea. Typically there's blood. Most often this is a dysentery, nausea, vomiting, and it can last up to weeks, but it's typically self-limiting. It'll just go away on its own. You can treat it with antibiotics though. Sometimes it's very mild and uncomplicated disease, but other times it causes appendicitis, actually. So here I wrote appendicitis-like disease, but we now know that Campylobacter can actually cause appendicitis. Rare complications include reactive arthritis and Guillain-Barré, which is sort of a over reaction of your immune response where you attack your own nerve cells, okay? There are vaccines to Campylobacter under development, which would really be great because, like I said, this is a very common diarrheal disease in the US especially. Like I said, Campylobacter jejuni is the big one. And if you look at it under a microscope, it has these helically curved rods, and they grow in pairs for some reason, and they look like gull wings, like a seagull, like seagull's wings. And they have a single polar flagella, and they're microaerophilic, and that's important to remember. In particular, they only like a small amount of oxygen. So when we want to identify, speciate Campylobacter, what we do is that we get a stool sample and plate it on plates containing this media called Campybac medium. And Campybac medium contains sheep's blood, and the hemoglobin in sheep's blood helps to suck up the oxygen surrounding the bacteria, keeping the oxygen low in the environment of the bacteria. And we can also put them in the plates in these bags that contain a reagent that sucks up the oxygen, keeps the oxygen levels low. And if you see any growth, it's from a species that does not like oxygen. And that could be indicative of Campylobacter. Campylobacter are a member of the Vibrionaceae family. And like the vast majority of Vibrionaceae family members, they are oxidase positive. So you can see they form this sort of purpley color when grown in the presence of the reagent that would indicate that they're oxidase positive. All right, so now I'm going to finish the lecture by talking about two types of Salmonella and then Helicobacter. So the first type of Salmonella that I'm going to talk about is Salmonella enterica subspecies enteritidis. Nobody calls it that. You can just call it Salmonella enteritidis. It causes gastroenteritis. It's found in us, in animals, birds, especially chickens, and in their eggs. And within a day of ingesting Salmonella, it colonizes our gut, causes diarrhea, nausea, vomiting, fever, abdominal pain. It's usually self-limiting, but in elderly people and in infants, dehydration and electrolyte imbalances can really be a problem from Salmonella. So sometimes they'll require IV or oral rehydration. But usually we don't use antibiotics to treat Salmonella because it prolongs the carrier state. That is, Salmonella can set up shop in our body, as I'll describe for you in a moment, especially in the case of typhoid fever, and live forever in our gallbladder. That's for typhi, but we'll get to that in a moment. So Salmonella enteritidis, again, we get it from ingesting it. It has to absorb to our cells, and then it can actually migrate from our epithelia to the lamina propria. The layer of the mucosa called the lamina propria. It can actually multiply in our lymphoid follicles in our intestinal tract, but it is confined to the GI tract. This is not a systemic disease like Salmonella typhi, like we'll talk about in a second. The mechanisms of pathogenesis of Salmonella enteritidis are very unclear. We know that it has a type 3 secretion system, which is like a needle that it has to deliver its virulence factors into the cells in our gut epithelia. Somehow, Salmonella causes inflammation, the recruitment of our cytokines and prostaglandins to the area around the infection, and that can lead to dysentery when our epithelia, or colonic epithelia in particular, is attacked by our immune system, and in combination with the cellular destruction caused by the Salmonella itself. And Salmonella somehow also stimulates cyclic AMP production, leading to fluid secretion, electrolyte imbalance. But I will not hold you responsible for the mechanisms of Salmonella enteritidis pathogenesis, because it's just very poorly understood. Okay, but it does cause diarrhea that is typically bloody. It's typically a dysentery. Okay. Here's an electron micrograph of Salmonella enteritidis. You can see it has this really incredible flagella situation going on, highly motile. And the one important thing to remember is that on McConkey lactose plates, the traditional McConkey plates, the colonies appear uncolored or white. So E. coli, this is very important to remember, E. coli and Salmonella are very hard to tell apart. But one way that we can tell them apart is the fact that most E. coli are lac positive. Salmonella, on the other hand, are lac minus. Now, how the heck do we tell Salmonella apart from Shigella? Well, remember, we have this reagent called hectoin, right? And Salmonella produces hydrogen sulfide gas. And because it produces hydrogen sulfide gas, it causes the hectoin reagent to precipitate on these plates and forms this black gunge on the plates. And like all Enterobacteriaceae, the Salmonella enteritidis is oxidase negative, cannot use oxygen as a determinant electron acceptor. Now let's look for a second at a comparison of the E. coli and Salmonella API strips. Remember, E. coli is lac positive. So here is the reagent that shows whether bacteria can use lactose as a carbon source. And yes, E. coli can, and Salmonella, no, it cannot. And then let's look at this reagent here, H2S. Okay, does it produce hydrogen sulfide gas? These cupules here in the H2S row, they contain the hectoin reagent. And you can see here a green cupule or a black precipitate here. And the reason for that is that Salmonella produces hydrogen sulfide gas. However, E. coli, which does not, similar to Shigella, the cupule is not dark, not black. Okay, so that's how these remarkable strips work. You could still set these up by hand, but these days, this has all been like highly miniaturized and highly automated. So robots can set this stuff up now, like in a clinical diagnostic laboratory in a hospital, for example. Okay, ignore this. There's no table 3. I just want you to know that there are other agents of blood diarrhea and dysentery other than ones that I told you about. Enteroinvasive E. coli, Vibrio parahemolyticus, Flanificus, even other species that we didn't get to at all, Yersinia, for example. And not just are there other bacterial agents there that are gram-negative, there's gram-positive bacteria such as Staph and Clostridia and Bacillus. I'm sure you're familiar, at least to some degree, with Clostridium difficile, which is C. diff, which has become a real problem in modern times. C. diff is a bacterial disease that can kill you, and is often a problem when we take antibiotics that wipe out most of our normal flora. We're repopulated with Clostridium difficile, and it can kill us. So there are also non-bacterial agents of blood diarrhea or dysentery, parasites, and various viruses. So don't think that just because I focused on the bacterial GI pathogens, that those are the, and gram-negative at that those are the only agents. There are others. All right, skip this slide. I'm just going to finish by talking about typhoid fever, and then Helicobacter. All right. So typhoid fever is caused by Salmonella enterica, subspecies typhi. Everyone just calls it Salmonella typhi, and so I. It's exclusively a human pathogen. We don't find it anywhere else, not in other animals, not in birds. At least none of them get the disease that we have from it. So it's a terrible clinical picture, a very lengthy one. It's a systemic disease with a very slow onset. In week 1, the patient is lethargic, has spikes of fever, very tired, has like lots of aches and pains, incredibly constipated. This is coincident with a Salmonella typhi penetrating the intestinal wall. By week 2, the patient has a really high fever. Typically, they're delirious. They often have these rose spots on their abdomen, these red spots that form on their abdomen as the organism invades the bloodstream and the biliary tree. Now, not all people that have typhoid fever have rose spots, but if you see rose spots, you should think typhoid fever. It's a very common indicator of typhoid fever. By week 3, there's diarrhea and fever. And then by week 4, the fever subsides and the patient's exhausted because they've been through, like up to a month of this craziness. All right. So the part that I just described for you is this period here, this like up to 1 month period where the Salmonella typhi first encounters the small intestine, then they can move through our lymphatic system to the mesenteric lymph nodes, and there from the thoracic duct, in our neck, to cause a transient bacteremia in our blood. Macrophages that pick up Salmonella are doing the Salmonella a favor, because Salmonella grows really well inside of macrophages, can also grow in our liver, our spleen, our bone marrow. It's just a very well evolved bacteria to the human host. Then it could also move from our intestines in the bile to the gallbladder. So Salmonella absolutely loves bile. So in the gallbladder, it cannot be happier. And then here we can set up what's called cholecystitis, or the carrier state, where Salmonella can basically live indefinitely in the gallbladder in some people. And then from the gallbladder, it can move into the small intestine and cause ulceration of the pyre patches, which can cause diarrhea, hemorrhaging, dysentery, and perforation of the small intestine. And then once this is in the small intestine, it can be excreted back out into the environment, infect other people. Okay, so it's a big mess with Salmonella typhi. And what we look for in Salmonella typhi is what's called the VI antigen. So the VI antigen is a capsular antigen. So this is a cell surface antigen on the bacteria. And here I'll draw VI. So the VI antigen is on this axis here. And we see throughout the course of infection, the VI antigen ultimately reduces, goes away. However, in a patient that has Salmonella in their gallbladder, where the carrier state has been established, the VI antigen will remain high, until the patient is treated with antibiotics that are specific to that species or that strain of Salmonella typhi. You have to make sure that strain does not have resistance to the antibiotic that you're giving. Otherwise, you can actually drive the formation of the carrier state. The alternative is also a cholecystectomy, that is the removal of the gallbladder. Okay, and like I said, we actually monitor for this VI antigen using antibodies, various assays to that capsular antigen. Okay, so as I said, it has a capsule. That's one of its virulence factors. The capsule is antiphagocytic, but really salmonella are very happy inside of macrophages. The capsular antigen also appears to be a virulence antigen. I think that's why it's called the VI antigen, but it's also what we monitor. It has flagella, so we can monitor its H antigens, but it also has this funny way of switching around the gene that it expresses that encodes for the protein in its flagella. So the H antigen can actually shift specificity. So that's called phase variation. It has some cell invasion machinery. Again, I mentioned it has the ability to survive inside of macrophages. It triggers this fever that I mentioned, this inflammatory response because it has endotoxin, and it has a type 3 secretion system for injecting various virulence factors into host cells. So, like I said, it's a very well evolved human pathogen. So, the way we prevent Salmonella typhi from spreading in the community is to eliminate the carriers. I'm not saying kill the carriers. I'm saying, cure the carriers so that there's no people carrying it. Because usually you can tell if somebody has it actively if they have symptoms, but the carriers, sometimes they look perfectly healthy, which is really weird. But the classic or most famous example of this is Typhoid Mary. So Typhoid Mary lived about 100 years ago. She was a chef. She would travel from place to place, mainly working for very wealthy families, but she was a carrier of Salmonella typhi, and she knew she was a carrier of Salmonella typhi. And eventually, the health department tracked her down, figured out where people were getting sick from, and they told her that she had to be treated, and she refused. And they told her, well, you could refuse, but you cannot cook for people anymore. And she continued to do that, and she wouldn't wash her hands. And she spent the last years of her life in prison because she just refused to stop working as a chef. She would, work under pseudonyms and, infect people. And she's thought to be responsible for a number of deaths. Okay, so there are vaccines for Salmonella typhi. They last up to 5 years, provide protection for up to 5 years. And believe it or not, these vaccines are based on the VI polysaccharide. So it's the purified version of the polysaccharide that's used as a antigen for the vaccine. All right, skip over that. All right, now I'm going to conclude by telling you about Helicobacter pylori, which causes duodenal and gastric ulcers. So this is really a modern medical miracle, the story of Helicobacter and its association with gastritis and ulcers. So Barry Marshall and Robin Warren were two physician scientists studying gastritis and ulcers. And what they found is that whenever they took biopsies of patients that had these symptoms, that there was always Helicobacter pylori present. And they had the hypothesis that Helicobacter pylori may cause gastritis and ulcers. And everybody said that they were crazy because something like half the human population on the planet has Helicobacter pylori living in our guts, but nowhere near half the population has gastritis and ulcers. But they wanted to test this hypothesis. So Barry Marshall treated himself with antibiotics, made sure he didn't have Helicobacter pylori, biopsied himself, to confirm. Then he drank Helicobacter and gave himself gastritis. And for proving Koch's postulates and that Helicobacter pylori causes gastritis, and ultimately it's been proven that they cause ulcers, Robin Warren and Barry Marshall were awarded the Nobel Prize in 2005. And I could tell you that this was really a miracle, because prior to to proving that Helicobacter was the cause of ulcers, people would die of ulcers or they'd live in misery. There was no great, cure, especially back then, or treatment back then for ulcers. So Helicobacter pylori is a gram-negative curved rod. So it's you could think of it as a Vibrio neciae family member, if you'd like. It has these polar flagella. It's microaerophilic, consistent with the fact that it grows in the stomach where there's not much oxygen. We are the reservoir for Helicobacter pylori. For a lot of people, Helicobacter pylori is just part of our normal flora, and we'll never get a peptic ulcer or have gastritis. But it is the causative agent of chronic gastritis and peptic ulcers. And it's also a risk factor for gastric adenocarcinoma, as well as non-Hodgkin's lymphoma of the stomach, okay? So you cure people of Helicobacter pylori, you cut down non-Hodgkin's lymphoma and adenocarcinoma, and you can cure them of their peptic ulcers. And I could tell you momentarily how we do that. Excuse me for a second. Sorry, I had to tell my kids to keep it down. All right, so there are numerous virulence factors in Helicobacter pylori. They have these flagella that enable them to move through the mucus layer. They produce urease, which is an enzyme that they use to convert urea to ammonia, and this helps buffer the acidity of the stomach. They have adhesins that enable them to recognize carbohydrate antigens on the surface of our stomach. They have a number of other virulence factors, but the only one that I'll ask you to remember is this vacuolating cytotoxin, otherwise called VAC-A. And what VAC-A does is it causes cell death by dysregulating our mitochondria. So in the endothelia of our stomach, VAC-A causes cell death by messing up our mitochondrial function. All right, so this is here just to recap that the Helicobacter must be able to survive this low pH, high acidity environment of the stomach and swim through the mucus layer to gain access to the epithelia. Here they can cause damage by dysregulating our mitochondria and then causing inflammation, which can result in an ulcer. Okay, so again, the symptoms are acute gastritis, sometimes diarrhea, but in the most serious cases, chronic inflammation of the gastric mucosa resulting in ulceration. So we diagnose this using gastric biopsy. We can isolate the bacteria, and then we can use a test for the presence of that enzyme called urease that converts urea to ammonia. So what we do is we can feed the patient radiolabeled urea, and we can monitor their breath for the presence of radiolabeled ammonia, because there's no way that you're going to produce radiolabeled ammonia if someone feeds you radiolabeled urea unless you're producing urease. And the only way you're going to produce urease is if you have a Helicobacter infection. So we can cure Helicobacter with a 3-drug combination. A common one is amoxicillin, and then another antibiotic, as well as a proton pump inhibitor to reduce the acid level in the stomach. All right, you can review this on your own, just sort of key concepts of everything I just described. You can review the objectives on your own, and I'd just like to finish with one more example question. Okay, a 4-year-old girl presents to the ER with a 2-day history of severe diarrhea that has become bloody. She has abdominal cramps and recently ate peanut butter sandwiches. She attends a daycare center 4 days per week. Her stool culture reveals straight gram-negative rods. What single additional finding would identify the cause of her dysentery? All right, so straight gram-negative rods. So we know that that means it's not a Vibrio neciae family member. So knowing that they're non-halophilic doesn't help us because all of the Enterobacteriaceae are non-halophilic. Same thing for oxidase minus. Lactose non-fermenting might help us a little bit, but that's not going to be like a single additional finding that's going to tell us what it is. But if we know that it's hectoin positive, then we know it's Salmonella and not Shigella. So this would be the right answer here, Salmonella and hectoin. So, and then knowing that they have a fever wouldn't help us anyway, because that's very common with dysenteries. Okay, so something like this, but you know, I, especially on an exam test, I make sure there's no ambiguity. All right, that's it. You know, it's very tough to avoid these GI pathogens because if you ever eat out, you don't know how people are treating the food you eat before you eat it. And that's just the risk we run, you know, living in a modern society where, you know, somebody else is, you know, growing our vegetables for us and, you know, in a lot of cases, you know, producing our eggs and cooking our food. So good luck out there. And if you have any questions, I'll be happy to take them. All right, I think we're good, Ian.
All right. Thank you very good. Thank you very much, man.
Thank you.
I think if I stop my recording, it stops yours. So.
Okay, yeah, I'm trying to be careful what buttons I press here. I don't want to do the wrong